Hey guys, hopefully you all had a nice weekend and happy Easter if you celebrate it. So I'm sure you guys noticed that there are like a lot of cell types and like transporters and channels that you have to know for the kidney. I found my notes from last year and I had made like a one-page note that has like all of the cell types and like all of the channels and transporters that are found in different regions of the kidney. So I'm going to remake those for you guys and post them so you guys can see everything kind of put together, probably over spring break because that's probably when I'll have time to do that. And hopefully you also saw that Dr. Yip posted that countercurrent multiplication video. So make sure to watch that as well because it's super helpful in being able to visualize like what's going on with that because that was kind of more one of the more like complicated concepts to really understand. So if nobody has any questions, I'm going to go ahead and start. We're going to do lectures 2 and 3 today. That way we're caught up. So that next week we only have to do 2. All right. So I'll go ahead and start then. Okay, so he's going to go into all of these in greater detail. So as he goes through all of the kidney lectures, so you don't have to like necessarily memorize these because you'll learn about them in greater detail as we go along. Okay, so you do have to know how like blood flows to the kidney because it's important in being able to understand all the other concepts. So you'll note that the blood entering your kidney is going to come in through the renal artery. The renal artery will separate into multiple other things that we don't need to know, but ultimately that blood is going to make up the blood as that goes into your afferent arteriole. Your afferent arteriole is then going to go into your glomerulus, which is going to break into your glomerular capillaries. And this is where you're going to see filtration of fluid into the kidney tubules. So not all of that blood enters as plasma and gets filtered out. The rest of it leaves through the efferent arteriole. So that would be this one here in this picture. And that efferent arteriole will then break up into separate capillaries. The ones that we need to know are your vasa recta and your peritubular capillaries. So your peritubular capillaries are going to be these right here that are going to kind of surround the tubules and help with reabsorption of substances that you reabsorb from your urine. And your vasa recta is going to be really important when we talk about countercurrent exchange, but it's going to kind of make a loop as well, and it's going to go down with the loop of Henle and pick up and drop off substances in that region. And then the blood that leaves the vein, the kidney, is then going to come out through the renal vein. So this is pretty much your entire blood flow through the kidney. One of the more important things to remember is that your efferent arteriole does give rise to your vasa recta and peritubular capillaries. So whatever is found in your efferent arteriole is going to also be found in your vasa recta and peritubular capillaries. And that's going to be important when we talk about PAH and inulin. So he doesn't really talk too much about the sympathetic aspect of the kidney. The only thing he really talks about is your afferent and efferent arterioles and how you can vasoconstrict those to minimize blood flow through the kidney. But you'll also see that you have juxtaglomerular cells. These are going to be the cells that respond to sympathetic activation and lead to the secretion of renin. Renin is then going to go through that like angiotensin pathway and ultimately lead to an increase in blood volume. And then the tubule also has sympathetic innervation to it. And I think what it does is it increases sodium reabsorption. But he never really talks about that, so don't worry too much about that. Okay, so this is all of your vocabulary words that you do need to know. You need to know the difference between filtration, reabsorption, and secretion because it can get a little bit confusing as to what's going on where. So you should know what these terms are referring to specifically. So anytime the word filtration is used, that's referring to what's going on in the glomerular capillaries, where your blood enters and, releases some of that fluid into Bowman's capsule and, it enters the tubules. So anytime you see filtration, that's what that's referring to. Reabsorption is different. This is going from the tubule back to the bloodstream. So you'll note that in your proximal tubule, for example, you're going to see a lot of sodium reabsorption, and that's leaving the tubular fluid and re-entering the bloodstream. So that's going to be happening from the tubules back to the bloodstream. Secretion is going to be the opposite. So this is the blood that exits out through your efferent arteriole and then breaks into your peritubular capillaries and your vasa recta. So this is going from those other peritubular capillaries being reabsorbed back into, being secreted back into your tubular fluid. So that some of the plasma that doesn't get filtered out at the glomerular capillaries can get filtered out more downstream. And anytime we're doing that, that's known as secretion. And then excretion, don't get it confused with secretion, is the fluid that actually leaves your body. So when you pee something out, that's going to be referred to as excretion. Okay. So you'll see this a lot, and you don't necessarily need to know the histology of the nephron. I think somebody's mic is on. But anyways, you don't need to know the histology of the nephron because you will have to learn it in histo if you haven't already. So this is actually a pretty handy slide for histo, not necessarily for our purposes. But anyways, you should know that the proximal tubule, which is this one here, is going to have the largest surface area because of this brush border. And that's important because the proximal tubule is going to be responsible for reabsorbing most of the sodium. And so it needs to have a large surface area to have all of those little transporters on the cell membrane. And it's also going to have a lot of mitochondria because your sodium-potassium pumps require ATP, so that having a lot of mitochondria is beneficial there. Your thin ascending limb, which is going to be this one, is mostly just going to have like water movement, and so you're not really going to need too much active transporter, and so therefore you're then going to see fewer mitochondria. But that's the only thing I would take away from this slide. Okay, so for glomerular filtration, you do need to know that the driving force is hydrostatic pressure. So the blood coming into the glomerular capillaries is going to push the fluid out into Bowman's capsule, and that's going to be the driving force for filtering out some of the plasma in the blood at the glomerulus. So you should know that because of that, you're going to have higher pressure at the afferent arteriole, and it's also like a thicker or like larger diameter. So this is your afferent arteriole that's coming in at a high pressure, and it's going to push some fluid out. The blood leaving at the efferent arteriole is not going to be as high pressure. It's going to drop a little bit, like by 2 points, but you'll also note that it's also thinner, and that's mostly because you're losing some of this fluid, so your hydrostatic pressure is going to drop as a result of that. But keep in mind that you're only filtering out the plasma, which is just like the fluid and any solutes from there. So you're not going to be filtering any albumin under normal conditions or any other types of proteins because they're too big to be filtered out. So if somebody has protein in their urine, you'll know that they have some kind of damage to their kidney because that's abnormal. Okay, so you should know what these three sieves in series are, and this is going to be the three things that your plasma needs to flow through to get into your kidney. So this is your glomerular capillary here, where your blood is found. And this area here is going to be Bowman's space. So as the blood is moving in through the capillaries, some of it is going to continue forward onto the efferent arteriole, and some of it is going to be filtered out. So it's going to have to go through your capillary endothelium, which is here that I colored in red. That's the endothelial cells that surround your glomerular capillaries. It has to go through this basement membrane, which you said is made of laminin, but that's not really important. And then lastly, it needs to go through Bowman's capsule epithelium, specifically these things called podocytes, which you do have to know. They are super important for being able to filter things out and only allowing certain things into Bowman's capsule. So if you have damage to these podocytes, bigger things can seep through, like proteins and things that aren't supposed to be there. And that's how come when you have proteinuria, it's due to the fact that these podocytes are, breaking apart. So you have larger space between them and things can seep through a little bit easier. And that's going to tell you that you have like kidney damage. And that's irreversible. So anyone who has kidney disease, kidney damage, it's likely due to the fact that their podocytes are breaking apart and more things are able to filter through. So filtration is going to be dependent on the size and the charge of the molecules. I believe only positively charged molecules, or mostly positively charged molecules, can go through, because I believe the basement membrane is going to be like negatively charged, so it kind of prevents like other negatively charged things from going through. But anyways, here's a picture of the slits between the feet of the podocytes that only allow certain things to go through. But if you know these three things and the direction in which fluid moves through them, you should be fine. And then I added this. I had this from my notes from last year to kind of give you a better representation of what that looks like. This is your podocytes. Your basement membrane is here. This is your capillary endothelium. And then your plasma proteins are going to continue through. They're not going to get filtered out unless you have like some kind of weird thing going on with your kidney. And it's going to be like your small positively charged molecules that are able to go through because your basement membrane is negatively charged. So it's going to repel negative charges and attract positive charges. Okay. So this should look familiar again. I think the main point of this slide is to reintroduce the fact that there is no net filtration force between the capillary and the interstitium, right? Because the difference between their hydrostatic and oncotic pressure is 3. So you're going to see in the next slide, we're talking again about your Starling forces. So if you'll probably remember this from the muscle capillary from block 1, how on the arterial side, because your blood or your heart is pumping the blood at a high hydrostatic pressure, you are going to have a really high hydrostatic pressure, and your oncotic pressure is going to be 25. So this oncotic pressure, once again, is due to albumin in your blood that is wanting to retain the fluid inside of the capillaries. This is not going to change at all because you're not losing albumin. You're not really gaining, more fluid or anything like that. You're not changing the concentration of the albumin. So it's going to stay consistent from the arterial to the venous side. However, because when you go from the arterial side, which is high pressure, to a low pressure venous side, you'll see that the oncotic pressure will exceed the hydrostatic pressure because the hydrostatic pressure drops so much. And this is because it is a high resistance system with slow flow. And I think there was something like vaguely remembering a question on something about high resistance, but I think it was like the low resistance. The next slide, I think. Something about this was important. Okay, so yeah, that's why during the Starling equation, we learned it even though it was wrong. You have reabsorption of fluid on the venous side and filtration on the arterial side in your skeletal muscle capillary. So this system is a little bit different compared to your glomerulus, which is so this is a more important slide and definitely something you should note. Because we don't really have to relearn the skeletal muscle capillary. It's just to like compare the two. Okay, so your afferent and efferent arterioles, you'll see that the hydrostatic pressure doesn't drop too much because we're still in an arterial. The reason why it drops a little bit is because some of this fluid gets filtered out into Bowman space. So you are losing a little bit of fluid, but it's not so much that you're going to see a large drop in your hydrostatic pressure. It only drops by 2 points. However, your oncotic pressure does increase a little bit because since you're filtering out some of this plasma, you're going to ultimately lead to an increase in concentration of the proteins and albumin that want to retain fluid. So your oncotic pressure goes up because of the fact that you're losing some of this fluid, and therefore the concentration of the protein increases as a result of that. However, you'll see that even though the oncotic pressure increases by 10 points, it's still not going to be greater than your hydrostatic pressure. So your hydrostatic pressure, which wants to push things out into Bowman's capsule, is going to always exceed the oncotic pressure, which wants to keep things in. So this is going to want to promote filtration of the plasma across the glomerulus. So memorize that it is a low resistance, high flow system. I do remember that being important, even though he just said it and he didn't have it on the slide or anything like that. Another number that you should remember is 125 milliliters per minute. Don't worry about 180 per day. This is really important to know that your typical glomerular filtration rate is 125. For your net filtration pressure, this is going to be the pressure that is wanting to push fluid out, and it's 10 to 12 millimeters mercury. You can memorize this if you want, but I don't remember this one being as important as this one here. Okay. Also keep in mind that this is another pressure that you have to understand. So this is the pressure in the Bowman's capsule. So if you have like fluid in your Bowman's capsule being building up due to some kind of blockage or whatever, some kind of issue with your kidney, this hydrostatic pressure is going to want to prevent more fluid from entering because it's already full. So under normal circumstances, it's only 15, which isn't so bad. But if this gets higher, it's going to want to prevent filtration from the glomerular capillary, which is not what you want. And then the oncotic pressure of the Bowman's capsule is equal to 0 because you typically don't have plasma or proteins entering Bowman's capsule. So you should have no proteins or albumin or anything like that. That's going to give it an oncotic pressure. So under normal conditions, it is also 0. I also added this slide from my notes from last year. This is just kind of a flow chart of everything that influences GFR. So here's your net filtration pressure. So this is the pressures forcing things out of the glomerular capillaries. So this is your hydrostatic pressure of your glomerular capillaries and the colloid osmotic pressure of your Bowman space. So if your Bowman space had plasma proteins, it would want to promote movement of fluid towards the Bowman space, but that's usually 0 because you shouldn't have any proteins in your Bowman's capsule. So it's not really taken into consideration. And then the pressure wanting to pull things back in, I color-coded these in purple and blue. So this is what wants to keep fluid inside the capillary, and that's going to be your capsular hydrostatic pressure, so the pressure inside of the Bowman space, and also your colloid osmotic pressure, which is the pressure due to the albumin inside of the capillaries. So your KF value is also something to take into consideration, which is it is going to be dependent on your surface area. And that's, if you have a small surface area, you have a lower GFR, a large surface area is going to have a higher GFR. And then the permeability of your glomerulus. If your glomerulus is super permeable, you'll have a higher GFR because it's going to promote fluid movement into the Bowman's capsule. And then a low permeability would do the opposite. And he pretty much said that there are certain toxins that'll do this. So he could potentially use that in a question stem or something. Okay, so do memorize this formula. Here's that KF value that we already discussed, which is based on the permeability and surface area. So surface area specifically is going to be dependent on your mesangial cell contraction or relaxation. So these are type of muscle cells within your glomerulus, and they're going to respond to these different molecules. So your atrial natriuretic peptide, which is ANP, and nitric oxide are going to cause relaxation of the cell. So when you have a relaxation of the cell, it's going to increase the surface area, and that's going to increase the KF value. Angiotensin 2, endothelin, norepinephrine, epinephrine, and antidiuretic hormone are going to want to contract the cell. So you contract it, you decrease the surface area, you decrease the KF value, and this is going to decrease your GFR. And it makes sense, since we've already kind of learned about ADH, why you would want to decrease the GFR under the presence of antidiuretic hormone, because if you're trying to conserve water, you want to filter less water out so you don't have to reabsorb as much later. So antidiuretic hormone works to contract the mesangial cells, decrease the KF value, and therefore decrease the GFR. Okay, so this is just restating what we already discussed about the KF value and how it's dependent on its permeability and the surface area, and how increasing both of those will increase the KF value, which will increase the GFR. Okay, so he had this question here: which of the following would promote an increase in GFR? So we know that if you were to decrease your blood pressure, that's going to decrease your hydrostatic pressure. And if you decrease your hydrostatic pressure, this is that part of the formula. So here's your KF value. This is your hydrostatic pressure of your glomerular capillaries. This is the hydrostatic pressure in your Bowman's capsule. And this is going to be the oncotic pressure of your glomerular capillaries, which is dependent on albumin. So if you decrease your blood pressure, that's going to decrease this value here. And mathematically, that would show you that you're going to decrease your GFR. So that can't be the correct answer because you're decreasing GFR. Increasing plasma albumin. If you increase plasma albumin, you're going to increase your oncotic pressure, which is this value here. Since it's being subtracted, if you increase this value, you're going to decrease your GFR as well. So that's not the correct answer. And then increasing norepinephrine is going to decrease your surface area. As we saw before, that norepinephrine and epinephrine are going to lead to contraction of those mesangial cells, and that's going to decrease the surface area and represent like a lower KF value, which is also going to decrease your GFR. So if you decrease renal sympathetics, which is the opposite of increasing norepinephrine and epinephrine, that's actually going to increase your surface area because you have less of these floating around, causing the contraction in the mesangial cells, and so your KF value is able to increase. Okay, so do you know the clearance formula? So this is going to be the volume of plasma that is from which a substance completely removed in the kidney. So this is going to be your urine flow. So how much urine is flowing out of your body at a given time. I think it's like milliliters per minute. Your urine concentration of the substance that you're clearing out is this one right here. So if we're using like PAH or inulin, it would be the concentration of inulin or PAH in your urine, which is shown here. And then this is going to be your plasma concentration. So how much you start off with of this substance, or we're talking about inulin, then it would be inulin. So make sure you know this formula. And then CX is clearance of whatever substance we're talking about. He'll usually use it in milliliters per minute. Okay, so what we do need to know regarding this is inulin. Inulin is a good example of where we need to know the clearance formula to measure GFR. So inulin is special because it is something that is only going to be filtered out at the glomerular capillaries. So it's not going to be reabsorbed downstream, so you're not going to see it exit the tubule and re-enter your bloodstream. It's not going to be secreted, so the little bit of inulin that makes its way to the efferent arteriole and then down to the peritubular capillaries is not going to be filtered out at the peritubular capillaries. It's not metabolized, so we're not going to use it in our body and therefore decrease the concentration and throw off our results. And it's also not produced, so we're not making it. And so that's also not going to allow us to change the concentration in our blood and therefore throw off our results as well. And it's also not going to influence your glomerular filtration rate by, causing contraction of your mesangial cells or anything like that. So it's a good way of measuring your GFR because whatever you filter out is the amount that's going to be excreted because you're not reabsorbing it or secreting it or moving it across into your tubules or back into your bloodstream any other place aside from your glomerular capillaries. So it's going to be a really good indicator of your GFR. So we can use the clearance formula to measure your GFR when we are using inulin only. So this is only specific to inulin. You can't use a clearance formula for something else and have it give you the GFR. You can only do it for inulin. So you should memorize the clearance formula, and that for inulin, this clearance formula is going to be equal to your glomerular filtration rate. Okay, so this part is a little bit confusing. So I'm going to kind of walk through it a little bit slowly so you can kind of visualize it. Luckily, he's not going to ask you to figure one of these out on the exam or anything like that, but you should understand the concept behind it, and you should be able to walk yourself through it so you better understand the concept. Okay, so if we were to inject somebody with a certain concentration of inulin, in this case, our inulin is equal to 1. The concentration is 1 milligram per deciliter here. So that plasma and the inulin is going to start off with a renal plasma flow of 600 milliliters per minute. So this number is in milliliters per minute. So that blood and the inulin enters the glomerulus. Some of that plasma and inulin is going to be filtered out. So it's going to be filtered out in a 1-to-1 ratio. So inulin, the concentration of inulin does not change. It's still going to be 1 when it enters the Bowman's capsule. The rest of the plasma with the inulin, it's going to make its way down to the efferent arteriole. And you'll see that the concentration is going to stay the same because it's being filtered out in a constant 1-to-1 ratio with the plasma pretty much. So it's not going to change the concentration. Okay. So as this inulin is going down all of the tubules, your proximal and your distal convoluted, all of that, you're going to see some water reabsorption. So water is going to be reabsorbed, but inulin cannot be reabsorbed. So as you work your way down to the end of the tubule towards the collecting ducts, your inulin concentration is going to increase because water is being reabsorbed. So if you're removing water, you're removing that dilution, and therefore you're increasing the concentration of inulin. So at the end of it, your urine concentration of inulin is going to be 125 if your urine flow is 1. So this is going to be like milliliters per minute of urine flow. So some of that inulin that leaves the glomerular capillaries that doesn't get filtered out is going to be split into going down towards the peritubular capillaries, and the other half, or less than half of it, is going to go down the vasa recta. So you're going to have more renal plasma flow, so more blood flow through the peritubular capillaries because they make up a larger surface area. So most of your blood is going to go through the peritubular capillaries. A smaller portion, only 60 milliliters per minute, is going to be flowing through your vasa recta capillaries as well. But keep in mind that inulin cannot be secreted from your vasa recta or your peritubular capillaries. So it's going to go through these, nothing is going to happen, and it's going to come back out of the renal vein, and the concentration is going to drop to 0.8 because you are diluting it, reabsorbing water, that sort of thing. You can also get this value of 0.8 by adding the renal plasma flows of your peritubular capillaries plus your vasa recta capillaries, so 415 plus 60, and then your renal plasma flow that comes out towards your renal vein. We start off with 600, but because we lose some of that filtrate, it drops down to 599 because we're only losing 1 milliliter per minute in our urine. So when you put these two, add them together and then put it over 599, that's going to give you the concentration of inulin that re-enters through your renal vein that you don't filter out. So that's not as important as understanding that the amount of inulin that you do filter out at the glomerulus is going to give you the glomerular filtration rate. This number should look familiar. It's 125, which is our typical GFR. Okay, I think that's all I wanted to say about this. Okay, so because it would be more of a hassle to inject somebody with inulin and then wait a while and then measure the clearance of inulin from their kidneys, it makes more sense that we would want to measure GFR using something that we make endogenously, so something that our body already makes and secretes constantly. So creatinine is going to be this thing that we are going to be secreting constantly that is going to be cleared at a value that is very similar to our glomerular filtration rate. So it's going to be a good measure of our GFR without having to go through the hassle of injecting someone with inulin and all that. So you should memorize this formula, which is literally just the clearance formula again, except this time it's specific for creatinine. And you should know what each of these things represent, and that for creatinine, you can use that to measure GFR. I think somebody's mic is on. Okay. So here is a list of the typical clearance values. You don't have to memorize all of them, but you should memorize the ones that we talk about, specifically these three. Don't worry about urea or sodium or water. This is that 1 milliliter a minute that we lose in the plasma. So that's why our renal plasma flow goes from 600 going into the glomerular capillaries. And then through the renal vein, it exits out as 599 because we do lose that 1 milliliter per minute of water. But keep in mind that we're going to see zero under normal conditions of glucose and albumin. Remember, albumin is not something that's supposed to be filtered out unless you have some kind of issue with your podocytes, and then you'll start to see some protein in your urine. But under normal conditions, this should be zero because it shouldn't be filtered out to begin with. Glucose, on the other hand, gets filtered out at the glomerular capillaries. However, it is going to be completely reabsorbed in your proximal tubule. So it should also be zero by the time it gets to your urine. So creatinine we talked about, it's a little bit higher than your typical glomerular filtration rate. So it's not a perfect way of measuring your GFR, but it's pretty close to what it should be. So that's why it's just more convenient to do it that way. And the reason why it's a little bit higher because there is some of it that gets secreted downstream. So most of it gets filtered out at the glomerular capillaries, but some of it does escape out through your peritubular capillaries, and that's why it gets to be a little bit higher than normal. Inulin is a little bit better for that. It's more accurate, as we already discussed. And then PAH, which we're going to talk about, is going to almost all get excreted out. So it's all going to be excreted either by getting filtered out initially at the glomerular capillaries or secreted from the peritubular capillaries more downstream. And you'll see that that value is significantly higher. So it's going to be a really good measure of renal plasma flow or renal blood flow. Okay, so renal blood flow is different from renal plasma flow. When we're talking about plasma flow, we're talking about just like the fluid aspect of the blood. So renal blood flow, you'll see that like a lot of your cardiac output goes through your kidneys. So you're going to see a lot of blood flow to your kidneys. You can memorize these numbers if you want, but I don't think it's super important. Your renal plasma flow is probably more important to remember because that's something that we're going to be measuring using PAH. So your renal plasma flow is equal to the renal blood flow minus the volume of hematocrit that is in your blood. So you'll see that it drops down from going from like 1200 to 600. So you can pretty much notice that like your hematocrit makes up like half of the volume of your blood. Once you take it out, it drops down by half. So do memorize 600 to 700 milliliters. Okay, and then another formula or relationship you should know is your filtration fraction. Your filtration fraction is going to be a ratio that tells you how much plasma is being filtered out. So your renal plasma flow is the total amount of plasma that is going to your kidneys and has the potential to be filtered out. And your GFR is what actually gets filtered out at the glomerular capillaries. So this ratio is going to tell you how much of your renal plasma actually gets filtered out. And under normal conditions, it should be 20%. And you'll see that this is your GFR, which is normally 125, and then 650 is usually your average renal plasma flow.
Hey Phoebe, I'm
Yeah.
sorry. What's the difference between like renal plasma flow and GFR? That's just like, I guess, the blood flowing through the efferent arterioles?
Yeah. So, whatever blood enters your afferent arterioles, that's going to be your renal plasma flow. Your GFR is what gets filtered out into the tubule. So, it's always going to be less than your renal plasma flow, because you're not able to like remove all of the plasma from your glomerular capillaries. Does that make sense?
Yeah, and so PAH is just like it doesn't measure just GFR, it's going to be like secreted as well, which is why it measures renal plasma flow, I guess.
Yeah. So it's going to be used to measure renal plasma flow because of the fact that it's going to enter your blood and get excreted out entirely. So it's going to represent the amount. So the amount of PAH that enters your kidney to get filtered out is going to represent the amount of blood that enters the kidney that can get filtered out. Does that make sense?
Yeah, I guess I'm just confused about why PAH would be a measure of renal plasma flow, but like inulin would be just GFR, because wouldn't the PAH just kind of be going through the glomerulus to be filtered out, or would it have to also be secreted, I guess? Did
It's
you,
also secreted.
and so that, okay. I,
Yeah, the difference is the fact that PAH is secreted downstream and gets like excreted in its entirety. So its concentration is going to go from its original concentration to zero in the blood, or close to zero in the blood. So whatever blood enters with a X concentration of PAH is going to filter out all of the PAH either at the glomerular capillaries, or it'll get secreted at the peritubular capillaries or whatever. And so you can measure the concentration of PAH in your urine as being equal to the amount that entered your kidney to begin with. So that's why it's a measure of renal plasma flow. Does that make sense?
yeah, so basically it's getting like everything but reabsorbed, whereas inulin's only getting, I guess, absorbed into the kidney or into the nephron through the glomerulus.
Yes, exactly. So that's why
Okay,
inulin is specific to GFR.
got it. That makes sense. Sorry about that.
No, it's okay. Okay. Um, okay, so you don't have to memorize these values either, but you should know the general distribution of blood flow through the kidney. So most of the blood is going to go to the cortex, followed by the outer medulla, followed by the inner medulla. Really easy to remember. But don't worry too much about like the percentages or anything like that. Okay, and with this, you won't have to know how to derive this, so don't worry about actually deriving this formula because that's a lot. But you should memorize this formula here in the box. And what this is pretty much saying is that if we are trying to measure the renal plasma flow, we're going to need to use something that you do not find any concentration in the vein. So whatever enters the artery, the renal artery, so whatever concentration of PAH enters the renal artery, the concentration of PAH leaving the renal vein should be zero or very close to zero because it gets entirely filtered out. And that's why it is a good measure of renal plasma flow. So it is measuring, so the amount of plasma that enters your kidney to get filtered out is going to be represented by the amount of PAH that gets cleared out in your urine because it gets filtered out entirely. So if you use this like clearance formula and you'll ultimately be able to derive it, you'll get this formula here, which is the clearance formula for PAH. And that is going to be equal to this thing called essential renal plasma flow. So because PAH isn't entirely cleared out, like it's mostly cleared out, but not entirely, this value is not going to be lower, I believe. Yeah, lower than what it should be. So the real renal plasma flow is going to be higher than this essential renal plasma flow, because PAH is not perfect. It's not a perfect measure, but it's like pretty close. So it's going to be equal, the clearance of PAH is equal to essential renal plasma flow. And then you have to divide it by 0.9 to get the actual renal plasma flow. So this is an important formula to remember. And so this is also very confusing. So I'm going to kind of like walk you through it to kind of get a better understanding of why PAH is a good measure for renal plasma flow. So remember, your renal plasma flow that is going entering your glomerulus is going to be 600 milliliters per minute. That's, the typical amount. And your PAH concentration in your blood in this situation is going to be 1 milligram per deciliter, I believe. So some of this PAH is going to get filtered out at the glomerular capillaries, and it's going to enter Bowman's space. And then the rest of this blood is going to make its way down to your efferent arteriole. Your efferent arteriole will then split into your peritubular capillaries and your vasa recta capillaries. So the concentration does not change because you're losing the same amount of plasma as you are of PAH. So it's like a 1-to-1 ratio. So the concentration doesn't change. Same with your Bowman space because you're losing plasma with the PAH. So the concentration of PAH is still 1. Because there are different amounts of renal plasma flow through these different capillaries, some of this blood, or most of this blood, is going to go down this direction through the peritubular capillaries, and then a smaller amount of this blood is going to go down the vasa recta capillaries. So the PAH that goes down the peritubular capillaries can get secreted back into the tubule. So this is your blood, this is in your body. It's going to get secreted out from the peritubular capillaries. So now you're going to notice that PAH is going to increase in your tubule, and it's going to decrease in your bloodstream because it's getting secreted out. So this is why it's not a good indicator of GFR, because it's going to mess with this value here. So the other bit of PAH in plasma that goes down the vasa recta capillaries is not going to be secreted. So whatever enters the vasa recta capillaries is going to stay the same. And so when it meets back up with the rest of this blood that has 0 PAH, because it's all been secreted into the urine, it's going to dilute it down, and your new concentration of PAH is going to be 0.1. Because this vasa recta does not have secretion of PAH. So whatever enters the vasa recta will leave the vasa recta in one piece. So this is why it's not perfect. It's not a perfect way of measuring renal plasma flow. So the value that you get by measuring the clearance of PAH is going to be your essential renal plasma flow. And because this is 0.1, if you subtract that from 1, you get 0.9, which is the value that you have to divide by in the formula in the last, this formula here. So you divide it by 0.09 because that's not exactly accurate. So that's how you get that. So your renal plasma flow, or your PAH concentration in your urine, is going to be equal to 540. So that is going to be the amount that gets filtered out here, plus the amount, your renal plasma flow of your peritubular capillaries here. So that's how you get the 540, because that's where you're getting your PAH from your peritubular capillaries, and the amount that gets filtered out. So it's going to be representing your PAH concentration in your urine is going to be representing the renal plasma flow at the peritubular capillaries plus your GFR. Okay, so that's how you get that value. And so when you divide 540 by 0.9, that value is going to be a lot closer to 600, or I think it's going to be 650 or something like that, and that's going to give you your renal plasma flow, like your actual renal plasma flow. So that's why you have to divide it by 0.9 so that this number gets a lot closer to the number that is supposed to be.
PBM.
Okay. Yeah.
So PAH, it behaves just like inulin until it gets to like later in the peritubular capillaries and the vasa recta capillaries. Is that right?
Right. Yeah. So inulin only gets filtered out here, but PAH can also get secreted out here. So that's the difference here. PAH will act like inulin in the vasa recta capillaries because nothing happens to it here, and neither does inulin either. But the peritubular capillaries is that difference.
And where's the dividing by 0.9 coming from again?
Yeah, so when you mix this blood here that didn't have any secretion of PAH, and you mix it in with the blood that got all of the secretion of PAH, so it should have no PAH, this concentration comes out to be 0.1. So if you subtract 1 minus 0.1, you get 0.9, and you divide by 0.9 to get the actual renal plasma flow.
Okay.
That makes sense?
Yeah, I think so. All right, thanks.
Yeah. Okay. So this is your typical PAH clearance value. So you'll see that it's slightly higher than normal. And so that would be your essential renal plasma flow. Okay. So just memorize that value and this value and this value, and the rest you don't have to worry too much about. Okay, so now we're going to talk about like auto-regulation of renal blood flow. So if you have normal blood pressure, so within a normal range of blood pressure, so 100 to about 200 or a little bit less than 200, your kidney is capable of maintaining constant blood flow by vasodilating, vasoconstricting, and in doing so is going to be able to maintain a constant GFR because your GFR is going to be dependent on the blood flow through the kidney. So if your blood pressure drops too low, your kidney isn't able to compensate for that, and so your blood flow is going to drop, and so will your GFR. And then the opposite is true for if you have a high blood pressure. If you have a high blood pressure, now your GFR is going to increase, and that's not a good thing either, because if your GFR increases, that increases the hydrostatic pressure, and you can actually damage your kidney, which is why a lot of people who have hypertension also tend to have like a renal disease because they are damaging their kidneys due to having a blood pressure higher than what is able to what your body is able to compensate for. So note that if it falls below 80 millimeters of mercury, you're not going to be able to maintain your GFR at a normal amount. Okay, so this slide is super important. So you should memorize what happens or understand what happens to your GFR, your renal blood flow, and I even included the filtration fraction, how that would change with changes in both of these. So if you were to constrict the afferent arteriole in this first example, you vasoconstrict it, and so you have less blood entering this afferent arteriole because you have more resistance. That is also going to decrease your GFR because now this is not, it hasn't constricted or anything. So you're not going to have any pressure building up here because you're having decreased blood flow and entering your glomerulus in the first place. So your hydrostatic pressure is going to decrease. Your GFR is going to decrease. Note that these two kind of function together. If your hydrostatic pressure decreases, your GFR decreases. If it increases, then your GFR increases. And renal blood flow is dropping because you have increased resistance, so you're letting less blood enter to begin with. So your filtration fraction will not really change, or if it would depend on the values for each of these.
Hey, Phoebe.
Yeah.
So this is like essentially the opposite of like what's going on in the body, because if you increase resistance, doesn't that increase blood pressure usually? So I would think that would mean you would filter more.
Okay, yeah, I see how you can. Yeah, so because you're like this, this region right here is staying the same, right? So even though you are like, there's going to be like more pressure pushing up in this region, and it could technically increase the hydrostatic pressure, but because this isn't changing in diameter, it's going to like ultimately lead to less blood entering this region. So you're going to actually see a decrease in hydrostatic pressure because there's less blood building up in this region because there's less blood flow overall. Does that make sense?
Yeah, because I thought earlier we had said that like if there was an increase in hydrostatic pressure, there's an increase in GFR. So is that really just talking about the capillaries when we talked about it earlier, or is that talking about the efferent and the afferent is different?
Okay, yeah. So if we're talking about hydrostatic pressure, we're talking about the capillaries because that's where you're going to see like the filtration, the fluid flowing out of. So this would represent your capillaries here. So that's not going to change in diameter. Your afferent and efferent are going to change their diameters in order to either like push more plasma through into the glomerulus or to like decrease the amount of plasma that's flowing through. So ideally, you want to increase the hydrostatic pressure in the capillaries by like, making it harder for blood to escape out. So then you have a higher GFR. Does that make sense?
Yeah, so essentially the hydrostatic pressure of like an increase in hydrostatic pressure of the either the capillaries or the efferents would increase the GFR, but an increase in the afferent would decrease the GFR. And when we were talking about it earlier, we were talking about just the capillaries.
Yeah, anytime we're talking about for the kidney, when we're talking about hydrostatic pressure, just think of it as for the capillaries because you're not going to see any fluid moving out at the arterioles. It's just going to be at the capillaries.
Got it. That makes sense. I'm sorry.
No, it's okay. Okay, so if you are now going to increase the resistance or vasoconstrict your efferent arteriole, so this is where the blood is leaving. By increasing the resistance, it's going to cause a fluid buildup because now it can't exit as easily as it could before. And so that's going to increase the hydrostatic pressure in these capillaries, and it's going to promote fluid flow outward. So that's going to increase your GFR. And your renal blood flow is going to decrease because anytime you vasoconstrict one of these, you're preventing how much blood can move across it. So renal blood flow is going to decrease. But that's good because if you have a high GFR and a low renal plasma flow or renal blood flow, it's going to increase your filtration fraction. Your filtration fraction is going to tell you how much plasma is actually getting filtered out. Okay. So if we were to dilate or vasodilate your efferent arteriole now, that's going to have a very similar effect to this one here because you are decreasing your hydrostatic pressure because there's no like buildup. So whatever enters can really easily leave because there's low resistance in your efferent arteriole. However, your renal blood flow is going to increase because you have a decrease in resistance. So if you're decreasing resistance, that's going to make it easier for blood to flow across. But your GFR is going to decrease because it is going to be dependent on your hydrostatic pressure in your glomerulus. And so for your afferent arteriole, your resistance, if you were to vasodilate, that decreases your resistance. So you will also have an increase in renal blood flow because it's making it easy for blood to enter your afferent and then go to your efferent. And because your efferent arteriole, it has a smaller diameter than your afferent arteriole, that's going to cause a little bit more blood to build up in your glomerulus, and that's going to increase your hydrostatic pressure, which will increase your GFR. And that shouldn't change your filtration fraction if they're both increasing by the same amount. So you'll notice that if you want to increase or decrease both your renal blood flow and your GFR, or increase both your GFR and your renal blood flow, you're going to be changing the diameter of your afferent arteriole. So that's kind of what our body does. It's going to mostly be changing the diameter of the afferent arteriole because that is like what is going to change the renal blood flow and the glomerular filtration rate in the same way. Okay, so you should know the two other mechanisms of autoregulation. Actually, he gives us like 5 examples, but these are like the main ones that you need to know. So tubuloglomerular feedback is going to be done by the macula densa, which I believe he'll talk about more when we talk about one of the last lectures, I believe. Okay, so anyways, your macula densa is going to have these specialized cells that are going to be picking up signals from the tubular fluid based on how much sodium chloride is in it. So if you have a high GFR, you're going to be filtering out more sodium and chloride, and that tubular fluid will make its way to the macula densa. And if it has high sodium and chloride, that's going to tell the macula densa that the GFR is high because you're able to filter out more sodium and chloride into the tubular fluid. And then that macula densa is then going to go and tell the afferent arteriole to vasoconstrict, so decrease its diameter, and that's going to decrease the GFR and decrease the renal blood flow. So it's going to decrease your GFR back to a normal amount. So this is just a way of maintaining a constant GFR. So you're not filtering out too much and you're not filtering out too little. So make sure you understand how your macula densa does this by measuring your sodium and chloride levels in your tubular fluid, so in your pee. Okay, so the myogenic response is very similar to what we talked about in Scalen's lectures, where if you have an increase in blood flow to your kidney, your vessels are going to recognize this as a change in pressure. So as your pressure against the walls, so if you have more blood flow, it's going to have greater pressure up against the walls of this vessel. And that's going to tell the vessel to vasodilate. And the purpose of this is to maintain constant blood flow to your kidney so that you don't overwhelm it with too much blood. So it's the same exact myogenic mechanism for autoregulation that we talked about in block 1. Okay, so this is a good summary slide of all of the different ways you can control renal blood flow. So blood pressure, we already talked about how you can maintain a constant GFR and constant blood flow. Given that your blood pressure is between those normal limits, so between 75 and 200, you can maintain it. If it goes above those, then you'll have issues with controlling your renal blood flow. Your myogenic and tubuloglomerular feedback we already talked about. But with prostaglandins, so prostaglandins is going to cause vasodilation. Sorry, my notes are kind of cryptic here. But this is going to cause vasodilation if for whatever reason your renal blood flow is really decreased. So if your renal blood flow decreases a lot, this will cause your vessels to vasodilate so you can get more blood flow through the kidney to get it back to normal. And then your sympathetic stimulation, remember when you have sympathetic, that's going to lead to vasoconstriction, and that's going to decrease renal blood flow. And then lastly, angiotensin, we'll talk about this again when we talk about the hormones. And that's going to lead to vasoconstriction to decrease GFR, and that's for the purpose of conserving water and, decreasing loss of fluid, loss of sodium, and that sort of thing. Okay, so does anyone have any questions about this lecture before I go on to the next? Okay, if not, I'll go on to the next lecture. Okay, so like I said, there's a lot of like little cells that you have to know and like the different transporters for different ones. So keeping like a list or something that kind of puts them all together based on what part of the kidney they're in or what part of the tubule they're in, that will help you like remember these. But it's really important to remember that your sodium and potassium pump is going to be important for maintaining your concentrations of sodium and potassium. So remember, sodium is going to prefer the interstitium because this pump is pumping out sodium. So you're going to have a low sodium concentration inside of the cell, and you're going to have a high potassium concentration inside the cell because it's being pumped in. So this is why when we talked about your compartment volumes and all of that, why potassium prefers the inside of the cell and why sodium prefers the outside of the cell, because of this pump here that your cells have. And so it forms this gradient to increase sodium in the ECF and increase potassium in the ICF. And so anytime sodium is entering the cell, it's going to be moving down its concentration gradient because you have low amount of sodium inside the cell, and it's high inside of the tubular fluid because you filter it out. And so it's going to be in higher concentration. So it's going to be moving down its gradient. And to get sodium out of the cell, it's going to be moving against its gradient. So that's going to require ATP and this ATPase. And it's going to move potassium into the cell. That's going to go against its gradient, and sodium out of the cell, which goes against its gradient. So keep that in mind. So most of the sodium reabsorption is going to occur in your proximal tubule, and so that's something important to remember about the proximal tubule. Another really important fact about it is that it's going to reabsorb things in an isoosmotic manner. So the osmolarity inside the tubule is going to be 300, and also on the outside of the tubule. Kaylin, this question is probably a little out of scope, but what kind of gradient is it if they're both positive? Is it just a concentration gradient for each cation? So I guess for that, it would be a concentration gradient for each cation. But for some of these transporters, you're moving like 3 sodiums out and 2 potassiums in. So because you're moving 3 positive charges out, you do see a little bit of a charge gradient there just because of the number of cations that you're moving out and in. Okay. Okay, where was I? Okay, yeah, and then this is the same exact slide that we saw in the first lecture where it's saying that you have a lot of mitochondria because it needs that ATP to be able to use for the sodium potassium ATPase. And then your brush border is going to allow it to have a higher surface area, so you have more of those little transporters that are useful if you have more surface area to use them on. So you're going to have a isoosmotic movement of solutes and fluid across the tubule. Okay. So you'll see this again. It's kind of a confusing table. You don't have to memorize these values, but you should understand what's going on and what these values represent. So if something has a high tubular fluid to plasma ratio, that means that you have a higher amount of solutes in the tubule compared to the plasma. So it means that something isn't going to be reabsorbed into your bloodstream. So it's going to either increase in your tubular fluid, or water is being reabsorbed, and so its concentration is increasing in the tubular fluid. If you have a low tubular fluid to plasma ratio, that means that whatever substance this is being reabsorbed back into the blood or into the interstitium. And so its concentration in the tubular fluid is less than its concentration in the plasma. And then anytime it equals 1, this means that there is equal osmolarity for both. So whatever is found in the tubular fluid, its concentration is equal to whatever is found in the plasma. And that makes sense because for a lot of these, you're going to see that the ratio is going to be 1. So this one, this one, this one. And that's going to indicate that you are reabsorbing these substances is in an isotonic manner, which is one of the main things to know about the proximal tubule. So sodium is going to be reabsorbed isotonically, potassium, calcium, all of that is going to be reabsorbed isotonically. You'll note that inulin, when it goes from the Bowman's capsule, so this is like right after it's filtered out in all of them are filtered out in a 1-to-1 ratio because your Bowman's capsule or your glomerulus is going to filter fluid out in an isotonic manner, always. The only thing here is that your protein, because it's not really supposed to be filtered out of your Bowman's capsule because protein doesn't typically, enter your urine, it's going to be lower because you're not supposed to have it in your you're not supposed to filter it out. But for the most part, all of them are going to be filtered out in an isotonic manner. So as you move across the proximal tubule, and by the time you get to the late proximal tubule, your concentration for inulin is going to increase. And this is because water is being reabsorbed in the proximal tubule. And we know that inulin cannot be reabsorbed at all. So its concentration is going to increase due to the loss of water. Sodium and potassium and calcium and the osmolytes are going to be reabsorbed in an isotonic manner. Chloride is different. I think for bicarb, we're going to talk about this when we talk about your acid-base balance in a later lecture, so don't worry about it yet. But chloride, that's going to be important for something in a couple slides from now. Okay, so these are the types of transporters that you'll find in your proximal tubule. So all of them are going to be co-transporters with sodium. So sodium is required to transport glucose, your phosphate buffer, your amino acids. This is going to be important when we talk about acid-base balance in the kidney. So don't worry too much about it yet, but for the purpose of just, introducing this concept, sodium is going to have an antireporter with with a proton, and that's going to help form CO2 and H2O, which can cross the cell membrane and then be converted into bicarb again and just, be excreted or reabsorbed or whatever. But anyways, this is your ATPase. That's super important for maintaining this gradient of potassium and sodium, so that potassium is high on the inside, sodium is low on the inside. And that's going to allow sodium to enter the cell down its gradient. So these don't require ATP. So he keeps changing the direction of which side is the blood side, which side is the P side. So if you are going to kind of draw out these cells, try to make them all on the same side so you don't confuse yourself, because it does get really confusing. So this is going to be reabsorption of sodium and all of these other things from the tubule. So this is your P side, and then into your blood. So this is reabsorption. He talks about this one in detail, so I would definitely know these transporters, their names, because he gave it its own slide, which probably means it's important. So sodium is going to help reabsorb glucose as well. So after it's filtered out at the glomerular capillaries, this urine is going to enter your proximal tubule. And so now you're going to start reabsorbing the sodium and glucose. So sodium moves down its gradient because there's less sodium inside the cell. So it's going to move down its gradient passively, and it's going to bring glucose with it. So glucose is then going to move down its gradient back into the blood passively through this GLUT2 transporter. And then sodium needs to get pumped back out because it's going to be moving against its gradient, because remember, sodium is high in the ECF, which is going to be your blood and also your tubule. But yeah, so it's going to get pumped out and potassium is going to get pumped in. So know which ones are passive diffusion and which ones are going to be like active transport across. Okay, so I thought this slide was kind of confusing, but pretty much what he wants you to know from this is that as your plasma glucose increases, so as your plasma glucose increases, you're going to be filtering out more glucose. So this is representing what's going on at your glomerular capillaries. So here's your Bowman's capsule. These are your capillaries. You're filtering out more glucose if your plasma glucose concentration is high, which makes sense because if you have more glucose in your blood, you're going to be filtering out more glucose into your Bowman's capsule. And then that, tubular fluid that enters is then going to go into your proximal tubule. And so you're going to want to start reabsorbing all of this glucose that you filtered out. So because reabsorption, as we just discussed, requires those transporters, there comes a point where they will become saturated. So you run out of transporters, and so you're going to start excreting some of this glucose because you're unable to reabsorb it. So At this saturation point, your excretion starts to increase because you're no longer able to reabsorb it like normal, and your reabsorption is going to plateau because you're going to not be able to increase the amount that you reabsorb. So you're going to be peeing out more glucose. So this is what happens to diabetics, why they have glucosuria, because this glucose is high in their blood and they cannot they filter out more of it, and that leads to increased excretion because their transporters are all saturated, so they wind up having glucosuria. And here is just like a little formula. I don't think it's super important to remember, but just know that whatever is excreted is going to be the difference between what is filtered minus what is actually reabsorbed. Okay, so going back to this table, and then it's kind of showing you what was in this table, but as a graph, which I think is easier to understand than the table. Okay, so your sodium and water that is being reabsorbed is going to be reabsorbed isotonically. So it's going to always be 1. So anytime it's 1, as we discussed in this chart, this means it's being reabsorbed in an isotonic manner. So because sodium is required to be reabsorbed with all of these other things like glucose and your amino acids and your bicarb, at the beginning of the proximal tubule, you're going to have mostly these things being reabsorbed until they go down to 0 because they're supposed to be 0. Glucose is supposed to be 0. Your amino acids are supposed to be 0. So you'll reabsorb them all in the beginning part of your proximal tubule. Inulin, its concentration is increasing because you're losing water in the tubule, and it's not being reabsorbed because it can't be reabsorbed. And then chloride is kind of weird because it is going to move with sodium towards the end of the proximal tubule. But at the beginning of the proximal tubule, since sodium is going with all of these other things, it's kind of ignoring chloride. So the chloride concentration at the beginning of the proximal tubule, since it's not being reabsorbed, it's going to increase its concentration because you're seeing more water reabsorption without chloride being reabsorbed. So this ratio is going to increase, but then once it gets to a particular region in the proximal tubule, that's when you're going to start seeing more chloride reabsorption because now your glucose amino acids and all these other things that were taking up the sodium and working with the sodium to be reabsorbed, now they're mostly reabsorbed, so now sodium can work on reabsorbing this chloride. So past that point, you'll start to see chloride being reabsorbed, not in an isotonic manner because there is always going to be a little bit higher since it did build up in the tubular fluid initially, but then it's going to be start being reabsorbed in a 1-to-1 ratio, which gives it this linear line like sodium because it's being reabsorbed at the same rate as sodium since they start running kind of together. So the main takeaway is that chloride doesn't get reabsorbed until the latter half of the proximal tubule, and that glucose and these amino acids and bicarb are going to be reabsorbed with sodium at the initial part of the proximal tubule, because you'll see that this is the proximal tubule length. So as you go deeper down the proximal tubule, you'll see less reabsorption of these and more reabsorption of chloride. So that's why this number is a little bit higher, because it does build up initially before it starts being reabsorbed isotonically with sodium. Okay, so this is the cell for the late proximal tubule. So this is the cell type that you'll see that is going to be responsible for reuptaking chloride. So there are 2 ways, and you should know these 2 ways in which you can reabsorb sodium chloride in the late proximal tubule. They can be reabsorbed through a transcellular pathway. So this is driven by the gradient. So remember, sodium is low inside the cell, so it's going to move in and then, export a proton, so it maintains the same charge. And then it's going to move down and out into the blood through the ATPase, which is seen in like almost all of these cells. And then chloride is actually going to move with formate, but it's moving in the same direction as sodium. And so what happens, because of the fact that you are creating this weird gradient, because you're moving chloride into the cell, and that is going to cause the lumen to become more positive because you're moving chloride out of the lumen. So it's becoming more positive, and you're moving chloride into the blood, so this is going to become like more negative in relation to this positive charge, so it's going to be 0. And so you're creating this charge difference between the lumen, which is where your P is, and the blood. And so this is going to actually promote sodium and chloride to move through a paracellular pathway. So sodium is going to move down its electrical gradient because it's positive. The charge is more positive in the tubular lumen because you're removing chloride. And because sodium is positively charged, it's going to want to go towards a more negative region. So it's going to move across the paracellular pathway down its electrical gradient because sodium is going to be high both in the lumen and both in the blood as well because it's an extracellular space. So it's going to be high in both. So it needs to move down its electrical gradient. Chloride, on the other hand, is going to move down its chemical gradient, so it's not really going to rely too much on the charge. But because there are higher amounts of chloride in the urine, it's going to want to go into the blood where it has a lower concentration. So you should know that most of the chloride and sodium is going to be moved through the transcellular pathway, which is going through the cell. But like 1/3 of it is going to be moved through the paracellular pathway down its electrical and chemical gradient. So make sure you know that, and also make sure that you know that formate is going to be moving in the opposite direction as chloride, because I think that comes back as something important later on, if I remember correctly. Okay. So because you're moving these solutes, so the solutes, your sodium and chlorides, water is going to want to follow. You're removing these solutes from the urine, so your osmolarity is going to be less in your pee and higher in your blood because you're moving these solutes across, so your osmolarity in your blood is going to be higher, and that's going to promote the movement of water with these solutes as well. So this is just showing you that water can move through transcellular or paracellular pathways and follows the sodium and chloride that was moving in the last slide. And then it also can move other solutes along with it, but he doesn't really talk too much about that, so don't worry too much about it. Okay, so this is a good summary slide to, understand the main takeaways of the proximal tubule. So you're going to be reabsorbing most of your salts and your waters, or in your water from your proximal tubule. And then all of your glucose and amino acids are going to be reabsorbed in the proximal tubule. And you should also keep in mind that your proximal tubule is going to reabsorb things in an isotonic manner. So its osmolarity inside the tubule and in the interstitium surrounding it is going to be 300 because that's the osmolarity in your body. So it's going to be maintained because it's reabsorbing water and solutes together in an isotonic fashion. Okay, so he's using this slide as like kind of just a slide that he throws in to show you what the nephron looks like. A transition slide, that's the word I'm thinking of. So don't worry too much about it. So now we're talking about the loop of Henle. So he shows this a lot, kind of looks like a snake, but here is your glomerulus. This is where your glomerular capillaries are, filtering out their fluid. This is your, all of this is going to be your proximal tubule. Keep in mind it's 300 because it's being filtered out and reabsorbing. You're reabsorbing things in an isotonic fashion, so it's all 300 here. So we already talked about that. Now we're talking about your thin descending limb of the loop of Henle. So this is the limb that is going to allow water to exit. So it's going to be permeable to water, but it's not going to be very permeable to solutes. So water can leave or enter, whatever, but solutes cannot leave or enter. And this is going to play a role in creating this osmotic gradient, which is super important. So note that the thin descending limb of the loop of Henle is important for creating this osmotic gradient. Okay, transition slide again. So now we're talking about the thick ascending limb, which is also super important in creating this osmotic gradient. It is the opposite of the thin descending limb. It is impermeable to water, but it is permeable to solutes, meaning it has these co-transporters that can pump solutes out into the interstitium. So you can think of these two regions as being opposites to each other. One of them moves water only, and the other one moves solutes only. Okay, so these are the special co-transporters that you need to know for the thick ascending loop. So it's going to be responsible for pumping sodium, potassium, and chloride out into the interstitial space. And what that does is it's going to decrease the solute concentration in the tubular fluid. So what's going to happen is the water or the tubular fluid is going to become more diluted. But because it's pumping out all of these solutes into your interstitial space, you're going to have a higher osmolarity in your interstitial space. And that's useful because now your thin descending limb can move water out to help dilute all of this extra solute that has accumulated. And as you move down, your tubular fluid is going to have an increased osmolarity because you are removing water from this tubular fluid. So it's going to be more, it's going to have more solutes to water. So less water, more solutes. So it's going to become more osmotic or hypertonic. So that's why you'll see that when you get down to the medulla at the bottom part of this loop of Henle, your osmolarity is going to be 1200, which is a lot greater than your 300 that you started off with because you're losing water in the thin descending limb. However, when you move back up, you're going to be removing solutes. So your osmolarity is going to decrease again, as it continues to move up. And so as your water gets removed, you increase osmolarity going down, and then as you go back up, you're going to decrease your osmolarity because you're pumping out these solutes. So that is what's going to create your osmotic gradient, such that your medulla has a high osmolarity of 1200, and that's going to be super important because it's going to be responsible for drawing water out at your collecting duct, specifically your medullary collecting duct. So anytime you're thinking about the medulla, you have to know that the medulla is going to be very osmotic. So it's going to have a high osmolarity, and that's where you're going to see the bulk of your water reabsorption, because your water from your collecting duct is going to be drawn out due to this high osmolarity in an attempt to dilute it. So if you haven't watched the video that he has about the countercurrent multiplier, make sure you watch it because it really lets you visualize this whole thing, because it's hard to visualize in these slides, and I think it's super helpful. Okay, so this is kind of a look at the thick ascending limb of the loop of Henle and these co-transporters that they have. So this co-transporter is going to be transporting your chloride, potassium, and sodium. And because it is moving 3 sodium out at the ATPase, you're going to have a negative cell potential inside of it because you're removing more positive charge than adding positive charge. And I don't know if he mentioned this year or if I just added this in my notes from something we learn later in another lecture, but this is going to promote the movement of sodium, calcium, and magnesium across this paracellular pathway because of this negative membrane potential. This might just be a thing that he talks about later and that I just added to my notes from last year. But I don't know, I remember it being important. So maybe just keep this in mind in case he didn't already mention it. Okay, yeah. So make sure you know this transporter. It's super important. Know what it moves across, and know that it's going to contribute to the countercurrent multiplier. And that you also have this ATPase, which is going to cause this negative membrane potential. But no water is going to be moving across this cell because the thick ascending limb is impermeable to water. Okay. So this is putting it all together. You should know the difference between your countercurrent multiplier and the other one, the countercurrent. Okay, well, we'll get to it. But this multiplier is going to be the thing that creates the gradient. So it's going to be the act of pumping out these solutes at the thick ascending limb, and that leads to water being drawn out at the thin descending limb to help dilute it. And then that winds up increasing the osmolarity inside your tubule so that by the time it gets to the bottom, it's 1200. It's going to equilibrate with the interstitium surrounding it. So the interstitium is going to be 1200 towards the medulla. And then as it moves back up, it's going to be pumping these solutes out. So then you wind up decreasing the osmolarity inside this tubule. And it's also going to promote water movement out. So by the time you get to the distal convoluted tubule all the way up here, your osmolarity is lower than your like average osmolarity in your body. So that, as it goes across the distal convoluted tubule, you are able to move out more solutes, and that's going to decrease it even more within the tubule. But yeah, if you understand that the thick ascending and the thin descending limb both play a role in the countercurrent multiplier and creating this osmotic gradient, you should be good because that is important. Okay. And so he used this as an example of your countercurrent exchanger. So don't get that confused with countercurrent multiplication. So countercurrent multiplier creates the gradient, and it's the action of those transporters pumping solutes out into the interstitium to promote water movement out from the descending limb. The countercurrent exchanger is going to be the action of the vasa recta. And the vasa recta is going to be responsible for maintaining this gradient. So you know that blood has to flow through the kidney, and specifically the vasa recta, which is going to give this loop shape. Your vasa recta has this loop shape. And what it allows it to do is it allows it to move through the interstitium, picking up, depending on where it is in the interstitium, whether it's at an area of low osmolarity where it will pick up more water and carry it down, or if it's in an area at a high osmolarity where it's going to pick up more solutes. But regardless, because it's a loop, it's going to make its way back, and it's going to deposit the solutes or water that it picked up in the first place back in the original location that it picked it up at. So it's going to help maintain the gradient. This is better than having just like blood flow moving in one direction that has the potential to pick up the solutes and carry them away permanently instead of depositing them back where it found it. So that's why it's important to have this loop, because it allows it to exchange with the solutes in the water with its environment and leave it back in the original location where it found it. So it allows for this maintenance of the gradient. And so that's what he was trying to illustrate here with this bath water, example. Okay, so yeah, here's your vasa recta. It's looking like a giant loop, and that's because of this whole countercurrent exchange mechanism, and helps it maintain the gradient without carrying the solutes or the water away and destroying the gradient, because it wants to maintain the gradient. Okay, so here is your vasa recta blood flow. So keep in mind that your vasa recta blood flow is going to move in the opposite direction of your tubular fluid movement. So your tubular fluid moves in this direction, and I made my arrows wrong here, so sorry. Your vasa recta blood flow is going to move in the opposite direction. And so that's going to actually allow it to pick up solutes or water at a lower osmolarity, move it down and deposit it back at a, like, pick up solutes here and drop them off here, or pick up water here and drop it off here. And so whatever, when it moves back up, it's going to then take the solutes that it picked up in, say, this location, and then drop it back off here. So it's not messing up the gradient. In fact, it's maintaining the gradient by exchanging with the environment and not allowing for the gradient to be carried away or, destroyed by some other blood flow. So, main point is that your vasa recta is important for maintaining the gradient, and that it's specifically going to be picking up sodium, chloride, and urea, and then dropping it back off, like as it moves along. But it's going to be in the same general location, so the osmolarity gradient is maintained. Note that urea is going to be a major player in creating this osmotic gradient. But I think he talks about that later in more detail, but just know that urea is involved as one of the solutes that make this gradient. Okay, transition slide. Now we're talking about the distal convoluted tubule. Your distal convoluted tubule is going to have these sodium chloride transporters, co-transporters, that is going to pump out more solutes into the interstitium, and thus decreasing your tubular fluid osmolarity from 150 to 60. So now you have very hypotonic urine inside of the tubule. And these co-transporters are also going to contribute to the countercurrent multiplier system by throwing more solutes into this interstitial space. So here is your cell for your distal convoluted tubule. Here's your sodium and chloride co-transporter. Then you're going to move it into the cell down its concentration gradient, and then you're going to pump sodium out and take potassium in through the ATPase as you normally do to maintain the gradient and pull more sodium and chloride into the cell. Okay, and so that's going to remove more sodium and chloride from your urine and make it more hypotonic. Okay, so yeah, there it goes from 150 to 60, and so by the time it gets to the collecting ducts, your urine is very hypotonic. So now it is the job of the collecting ducts to remove the water to make it hypertonic again. So your collecting ducts have two parts. You've got your cortical collecting duct, which is located in the cortex of the kidney, and your medullary collecting duct, which is located in the medulla of your kidney. So it's going to be in the more osmotic region. So you should know the differences between these two. So your collecting duct has two different cell types. It's got your principal cells, which are the ones that we're going to mostly talk about in this lecture. And then it's also got your intercalated cells, which are more relevant to the acid-base lecture. So they're going to be responsible for like reabsorbing bicarb. But more importantly, the principal cell. So the principal cell is going to be, allowing sodium and potassium to move down their gradient. So sodium is going to enter the cell and then be reabsorbed here. But the most important thing to remember about the principal cell is that it's going to be the cell in which ADH can act on. So it has the antidiuretic hormone G protein coupled receptors on it that allow for water reabsorption. So you're going to see mostly medullary or mostly principal cells in your medullary collecting ducts. So that tells you that most of the water reabsorption is going to be found in your medullary collecting duct. And that makes sense because your medulla has that high osmolarity that is better at drawing more water out of the tubule. So as you make your way down your collecting duct, you're going to see more and more principal cells towards the medulla, because the medulla is the best place to have water reabsorption because of that high osmotic gradient. Okay, so ADH is going to bind to the GPCR. You don't have to worry about all this like signaling pathway. Just know it's a G protein coupled receptor, and that it ultimately leads to water channels that are usually sequestered in this vesicle to then bind to the cell membrane at the luminal side, right? So this is the side that has your P. Your principal cells have aquaporins on their other side that they don't sequester anything. They're just always there. But if you can't move fluid or water into from one side, then these are not going to be able to function otherwise. So it's going to take water in, and then water is going to go out through these aquaporin to be then reabsorbed in the blood. So you should know that these are aquaporin 2 channels that are stored in a vesicle until ADH can activate the vesicle binding to the membrane. So without ADH, your principal cells won't be able to move water across them, and you won't have any water reabsorption. So ADH is super important for water reabsorption in your collecting duct. And by removing water from your tubule, you're going to create hypertonic urine. Okay, so this is your cortical region, this is your medullary region. Note that it's more osmotic down here, so it's going to be better at reabsorbing water at the medullary collecting duct, which is why you have more of those principal cells present and less of the intercalated cells present. Okay. So also keep in mind, and I think he talks about the actual mechanism of action for urea later, maybe. But keep in mind that urea is going to be found in your tubular fluid because it gets filtered out too. So as it's moving down towards the collecting duct and water is being reabsorbed, you're removing water from the collecting duct, so your concentration of urea is going to increase. So by the time it gets down to the inner medulla, your urea concentration inside the tubule is greater than the urea concentration outside of the tubule. So now it's going to be able to flow down its down its gradient and enter the interstitial space. And urea is super important for maintaining this osmotic gradient because it acts as one of the solutes that hangs out in this interstitial space and maintains that gradient. So it kind of upregulates itself in a weird sort of way. So by drawing water out, you draw urea out, and urea being in this interstitial space is going to allow for more water to be drawn out. So it upregulates itself, kind of. So keep in mind, urea is going to move out with water in the collecting ducts, in the medullary collecting ducts specifically, because it has to wait until the water gets reabsorbed and urea's concentration gets to a critical point where it's greater. And after that, that's when it's going to be reabsorbed, which is in your inner medulla. Okay, so this is a good summary slide. So make sure you understand the things that ADH does. So obviously it's going to allow for water reabsorption by adding those aquaporin 2 channels to the membrane. And then by adding the water channels to the membrane and, promoting water in movement and reabsorption, it's going to ultimately lead to urea reabsorption as well. Because of that relationship there. So it's going to decrease vasa recta blood flow. So this is super important because remember, if you have your vasa recta blood flow being too high, it could potentially pick up too many solutes and carry them away permanently, and that could disrupt the gradient. So by ADH decreasing your vasa recta blood flow, it maintains the vasa recta blood flow at an optimal level such that it's maintaining the gradient without picking up too many solutes and carrying those away, or picking up too much water and carrying that away. So that's why it would want to decrease vasa recta blood flow. And then another thing that it does, which he didn't really mention, is it increases the expression of these cotransporters, which if you make more of these, then you can, upregulate that countercurrent multiplier system and then increase the gradient and then just make it work a whole lot better. So it's going to upregulate the gradient overall. Okay. And so your high medullary and interstitial osmolarity, so this is that gradient where you have like a really high osmolarity towards the medulla, that's going to be due to your sodium and chloride being pumped out of your thick ascending limb, which is part of the countercurrent multiplier system. The low flow through the vasa recta, remember low flow, decreased flow, that's going to help maintain the gradient because you don't want too much blood flow through the vasa recta to that could potentially mess up that gradient. The countercurrent multiplication, which is associated with these being pumped out of your ascending limb, and also your thin descending limb, which, moves water across it, which also helps with the countercurrent multiplication. Your countercurrent exchange, courtesy of your vasa recta, maintaining that. And urea. So urea also is one of the solutes that gets pumped out into your medulla and helps increase the osmolarity towards the medullary region in your kidney. Okay, so this is a super important slide to really understand the relationship. So this is going to be your tubular osmolarity. So this is going to be the osmolarity of your pee inside the tubule. So you know that in your proximal tubule, it's going to be isotonic because it's constantly reabsorbing things in an isotonic fashion. So it's going to always be 300. When you get down to the loop, as it works its way down towards the medullary region of the loop, it's going to be very high osmolarity because you have more solutes than water down there. And then it's going to move its way back up. And you're going to see that these transporters are going to be pumping out solutes. So your urine osmolarity is going to go back to normal and then actually exceed normal and then be very hypotonic. So by the time you get to the distal convoluted tubule, your urine osmolarity is going to be about like 150. And so your distal convoluted tubule has those sodium and chloride cotransporters, so you're now going to decrease the osmolarity of your pee even more. So this is where it gets a little bit tricky. The osmolarity of your urine is now going to be dependent on whether you have ADH or no ADH. So if you have no ADH, you can't put those water channels on the cells, and then therefore you can't move water across to reabsorb it. So your pee is going to be hypotonic. However, if you do have ADH, you can reabsorb most of that water, and so your urine is then going to be very hypertonic. It's going to have a high osmolarity because you reabsorbed all of that extra water. Okay, so that's it for that. Does anyone have any questions about this before we do the Kahoot?
On the last slide, can you talk about why the volumes change?
Oh, are you talking about this right here?
Yeah.
Yeah, so I think it has to do with the fact that you're like removing water. So it's getting reabsorbed. So it's being taken out of the tubular fluid, which makes sense. So here, going down here, since it's water permeable, you're removing water from the tubule. So by the time you get down here, there's only like 25. And then you're not changing the volume here because it's increasing or decreasing in osmolarity because you're pumping out solutes here. You're not moving water. So by the time you get to this, it's going to be the same volume as before. It's only changing the amount of solutes. So from here, I guess you see a little bit of water movement from the distal convoluted tubule. So it drops down by like 5. And then you'll see a lot of water being reabsorbed if ADH is present, but none if there is no ADH present. Does that answer your question?
Yes, thank you.
Yeah, okay. Yeah, Patrick, I can explain the countercurrent exchanger again. Let me get to that slide. So it's kind of like confusing, but so this tubule is pumping out solutes, right? So your countercurrent exchanger is just going to pick up whatever it's, seeing. So if the osmolarity here is, so it's going from an area of high osmolarity to a low osmolarity. So because it's going from an area of 1200, this blood is going to be equalizing with the interstitium around it. Because remember, your capillaries are going to equalize with the interstitium. So it's going to pick up more solutes at this, high osmolarity region. And so as it moves up, it's going to go to like a lower osmolarity, so it's going to be more inclined to release these solutes. And by doing so, it's kind of maintaining a gradient because it is picking up some solutes and carrying it away, but it's not carrying it away like to a different location where there's no osmotic gradient. Instead, it's like dropping off the solutes back to like an area where you would find solutes. So it's kind of like maintaining the gradient, especially since it's where the blood is moving in the direction opposite to the tubular fluid. So specifically, it's going in this direction because your tubular fluid is going in this direction. So it will likely pick up more water up here because you have less solutes. So carry the water down and start to dump it in an area where you have higher amounts of solutes. So it's going to dilute this a little bit, but by the time it gets down here, it's going to pick up a bunch of solutes and then it's going to move back up and dump these solutes here. So by doing it in like a loop shape, it's not messing up the gradient. Whereas if you just had like one capillary moving straight through this, it's going to pick up, or if it was moving in this direction, it would pick up all the solutes here and then just carry it away. Does that make sense, Patrick? Yeah. Oh, I'm sorry, Inga. Yeah, I can go over the difference between the countercurrent multiplier and exchange. So the countercurrent multiplier is really just the actions of the thin descending limb and moving water out in response to the thick ascending limb pumping out solutes. So this is what makes the gradient, these two limbs either moving water or solutes out. Your countercurrent exchange is what maintains the gradient, which is the action of your vasa recta capillaries, picking up solutes and dropping them off a little downstream to kind of like equalize with the gradient and therefore maintain the gradient. Does that make sense, Inga? Or are you still confused? It is like a little bit confusing because the words are very similar. Okay. All right. Does anyone else have any questions? Because I'll start the Kahoot now if not.
What gradient what gradient are the vasa recta capillaries maintaining exactly?
The gradient that goes like from 300 to 1200. So this like your interstitial gradient. So it's this interstitial gradient that is responsible for like drawing fluid out of your the collecting duct. So you want to make sure that this like towards the medulla, it's 1200 all the time. So it's just kind of just maintaining it so that the solutes just, I don't know, I guess they get messed up because your interstitial fluid is a fluid. So by like picking things up and like dropping it off, it's just kind of like keeping it all in the same place because it does form a loop. So by dropping it off in this location, it'll pick it up in this location and carry it, yeah. Does that make sense? Or are you still confused?
I'm still kind of confused. I mean, I know that the countercurrent multiplier is like the main thing forming that because you have tubular fluid come down it comes down the thin, descending Henle, and it's not letting any solutes out there. So because it only lets water out, that makes it super hypertonic at the bottom of the loop. And then when the ascending thick Henle comes up, it's not permeable to water, but all of its solutes leave. So that increases the osmolarity there, right, in the interstitium. And then what the vasa recta is just kind of there, like, what is the vasa recta doing to the solutes there? Is it's taking them up?
No. Okay, so it's going in the opposite direction. Like the blood flow is opposite to the tubular flow. So it's picking up solutes that are being pumped out at this thick ascending limb, and it's bringing it down here and like depositing them there so that you can maintain like this,
okay.
maintain the like low, high osmolarity.
So it's bringing solutes, even more solutes to the bottom and the medullary portion.
Yeah, it's going to maintain it really, really osmotic down here.
Okay. All right. That makes more sense. And also one last thing isn't there like a specific point where tubular fluids is like called urine? Because I noticed like you and Dr. Yip both kind of like switch back and forth with it. Is there a point where you don't say tubular fluid anymore, just so like in case I see it in a question?
I honestly don't know, and I don't think he'd try to trick you like that. I just like assume whatever's excreted is urine, and I just call it all pee because technically it will become pee, but probably towards the end of the medullary collecting duct after all the water has been reabsorbed, then I guess it would be urine because it's ready to be excreted. But honestly, I don't know.
Okay. All right. Thank you.
Yeah. Okay. Yeah, okay, so there's a question. If the vasa recta comes from the efferent arteriole, how can it be going towards the glomerulus? Okay, this picture is kind of awful. I think it's just a problem with the picture, but yeah, don't worry too much about this picture. It's supposed to be coming from the efferent arteriole, and then it breaks out and breaks into a loop. But yeah, I think it's just this picture is kind of bad. Sorry if that's confusing. Yeah. Okay. Any other questions, guys? All right. So I guess I'll start the Kahoot, and if you guys think of anything, you can ask me after the Kahoot. Okay, I'm going to start it. Awesome. Yeah. Okay. So keep in mind your arterioles are not going to be moving fluid across them because they're arterioles. You'll only see movement of fluid through capillaries. So your peritubular capillaries are going to be responsible for reabsorption, whereas your glomerular capillaries are going to be responsible for filtration. Cool. Yeah. So epinephrine is going to decrease the surface area, decreases your KF, which also decreases your GFR. Okay, so PAH clearance is going to be a measure of your renal plasma flow, not your GFR, because it's going to be completely cleared out. So because it gets reabsorbed, it's taking into consideration the amount that gets reabsorbed at the peritubular capillaries. Therefore, it is a good measure of renal plasma flow and not GFR. Only inulin is a good indicator of GFR and creatinine, technically. Okay, so yeah, if you have an increased amount of sodium chloride sensed by the macula densa, that means you are filtering out too much sodium chloride. So you're going to tell your kidney to kind of tone down, decrease GFR back to where it's supposed to be. And it does so by constricting the afferent arteriole, because if you constrict the afferent arteriole, that's going to decrease renal blood flow and also decrease your GFR, which is the purpose of that. All right. Okay, yeah, so filtration fraction is going to be GFR over renal blood flow. So if you have an increased renal blood flow and a increased GFR and decreased renal blood flow, that's going to give you a increased filtration fraction. Okay, yeah, so inulin is not going to be reabsorbed at all, so its concentration is always increasing in the proximal tubule. Chloride isn't going to be reabsorbed until later down the proximal tubule, so its concentration is also going to increase in the early proximal tubule. So both of them are going to increase. Okay, so your distal convoluted tubule only has that sodium and chloride cotransporter that he discussed. Awesome, it's the opposite. It's the countercurrent multiplier that creates the gradient, and then the countercurrent exchange maintains it through the vasa recta. Okay, so it's going to increase the expression of sodium, potassium, and chloride transporters because it's going to want to upregulate the production of the gradient. And by increasing expression of those transporters, you're going to be able to pump out more solutes and make that gradient. It's going to decrease the vasa recta blood flow, not increase it, because if you increase your vasa recta blood flow, you run the risk of ruining your osmotic gradient because it could pick up too much solute and carry it away. Okay. Awesome. Yeah, it's the principal cells in the collecting duct that have the receptors for ADH. So I'll stick around for questions, and I'll also post some good videos on countercurrent exchange and multiplication to make it a little bit easier for you guys to understand.
Hey, Phoebe.
Yeah.
Quick question on the first lecture slide. I think it's like 26. Yeah, 26. You just had that PAH is slightly higher than normal. Wouldn't it be slightly lower because you still have that little bit from the renal vein?
Oh, from this lecture, I think I mislabeled that when I put my notes in. Yeah, it should be slightly lower than normal that you excrete.
Okay,
Yeah,
cool.
I
Thank you.
mislabeled that. Sorry. Yeah.
No worries. Thank you so much.
You're welcome.
Hey Phoebe,
Yeah.
while you're on this PowerPoint, could you review the slide right before this one again one last time?
Yeah, this one, it's super confusing. Okay, so you start off with a concentration, a certain concentration, and to make the math easy, he made it 1 of PAH. And so your renal plasma flow that enters your glomerulus is going to be equal to 600. So some of that plasma with the PAH in it is going to be filtered out. The rest is going to make its way down to the efferent arteriole, and then it breaks up into your peritubular and vasa recta capillaries. So PAH is able to be reabsorbed, or I'm sorry, secreted into the tubule. So whatever enters the peritubular capillaries will get secreted into the tubule. So even though some of it doesn't get fully filtered out at the glomerulus, the rest of it or most of it gets, secreted back into the tubule through the peritubular capillaries. So now you have the amount, which is 1 because it's being reabsorbed in a it's being filtered out in a 1-to-1 ratio with water, so its concentration doesn't change. And it's being reabsorbed here as well. And then because water is being reabsorbed, so it's being secreted, water is being reabsorbed, you're removing water from the tubule, so the concentration of PAH is increasing because of that. So this is going to be equal. So your concentration of PAH is going to be representative of the GFR, so the amount that got filtered out initially, and your renal plasma flow for your peritubular capillaries because PAH gets secreted from these as well. So that's why it's 540. The PAH that enters your vasa recta capillaries cannot be secreted or filtered out or reabsorbed or nothing. It's going to make its way down to your renal vein, and it's going to mix in with this blood that got all of the PAH reabsorbed. So by the time it mixes together, the concentration of PAH is only 0.1. So that is 0.9 less than what it originally started with. So that's why you get that number to divide it in the effective renal plasma flow formula in the last slide. But point is, you don't have any secretion of PAH in your vasa recta, but you get 100% secretion of PAH at your peritubular capillaries. So that's why your PAH is a good indicator of renal plasma flow because not only does it take into consideration the GFR, so the amount of plasma that enters your kidney, but it's also taking into consideration your peritubular capillaries. The only one it doesn't kind of account for is your vasa recta, but because your vasa recta is so like small in comparison, it's not really a big deal, and you can just divide by 0.9. Does that make sense, or are you still confused about something?
No, I think the only thing I'm a little confused on is just if it's 0.1, why do we have to do the subtraction? Like why don't we just use the 0.1?
Yeah, so you're measuring like clearance. So
Okay.
0.1, does that make sense?
Yeah, once you said clearance, so it's how much is being excreted out. Then that makes
Yeah,
sense.
yeah. 0.9 is being excreted out, and then 0.1 is staying in.
Yeah,
So
that's perfect. All right, thank you so much.
you're welcome.
Phoebe.
I had a question about that.
Oh, you go ahead.
Just quickly. I was wondering, like how important that the vasorectal portion of this is to know. Like, are we just needing to know that it doesn't get excreted? I'm sorry, that it doesn't get reabsorbed back into the tubule, or like, is it more important to know like the peritubular side of it? I was curious.
So you should kind of just know that PAH doesn't get secreted from the vasa recta, only the peritubular capillaries. Maybe that's the only thing to take away from this. And then maybe know that not a whole lot of blood flows through your vasa recta in comparison to your peritubular capillaries. But I wouldn't really worry about the numbers or anything like that.
Okay, that's what I was kind of wondering about the numbers and how everything was important. If it was just like a conceptual thing that we needed to understand.
Yeah, just know the concepts and like understand like where most of your blood flows through. But don't worry about the numbers or anything like that.
Okay, thank you.
Yeah.
So the one thing that's still kind of like racking my brain is with inulin and PAH. I guess I'm just confused on how they both go through the glomerulus and they both make it to the peritubular capillaries and the vasa recta capillaries. But how is the concentration of both of those staying one when you're changing the renal plasma flow and you're also changing, what's in Bowman's space? I don't know. Maybe I'm missing like a basic Gen Chem property or something. I don't know.
Yeah, no, that's okay. So it's just kind of like the concentration is dependent on like how much water is going with it. So you're moving it in like a 1-to-1 ratio. So even though you have less blood flow at your vasa recta, you're getting less in PAH or inulin also. So it's decreasing the actual amount. So say like there's like 20 molecules going down this way to 20 molecules of water as well. And then this one would be like 50 molecules of PAH with 50 molecules of water. So it's like a 1-to-1 ratio with the water that it flows with. Does that make a little bit more sense?
So it's only regarding the actual water in the solution.
Yeah, you can think of it like as like the concentration of it with the water that is in the blood.
But depending on which one you're talking about, do both of those have a higher amount going through Bowman's space to the tubule? Is that right?
Wait, repeat that.
So with PAH, for example, like there's more of it going through Bowman's space, right, into the tubule than there is going to the
So not so the most of the PAH is going to your peritubular capillaries because the amount that gets filtered out is only 125 milliliters per minute, and then your renal plasma flow is 415 milliliters per minute. You can think of it that way. So most like only 100 like a like equivalent to 125 gets actually filtered out. So most of it makes its way down to the peritubular capillaries. So the highest amount of PAH can be found in your peritubular capillaries, but its concentration is not going to change because it's flowing in equal amounts with the blood. But like you can find more molecules of PAH in the peritubular capillaries because that's where most of the blood flows through. Does that make sense?
Yeah, but with inulin, don't you find more of it like in the tubule because it gets freely filtered?
So you mean like in here?
Yeah, like it doesn't go to the capillaries like PAH does.
So it actually does go to the capillaries. Here, let me go back and show you. Because only a small amount, like 125 milliliters per minute, actually gets filtered out. So you'll find inulin in the tubule, and you'll also find it in the capillaries and the efferent arteriole, and that turns into the paratubular capillaries. But it can't move outside of the capillaries. So whatever's in these capillaries isn't going to be reabsorbed or secreted or anything. It's just going to stay in there. But because you are having increased water reabsorption from the tubule back to the capillaries, you're kind of diluting the inulin, which originally started off as like a concentration of 1, but because you're adding water, you're diluting it, so its concentration drops down to 0.8. Does that make sense?
So what do they mean by inulin is freely filtered then?
It's freely filtered at the glomerulus. So however much plasma is filtered out, inulin is also filtered out with it.
Okay. All right, I think I see it. And there's more renal plasma flow, so you end up with more inulin over in the capillaries at first.
Yeah.
Okay. All right, thank you.
You're welcome.
Hey Phoebe, I was wondering if you could go over the Starling courses again a little bit more slowly,
Yeah.
please. Thank you.
Yeah, of course. So you mean like these slides here?
Yeah, the one about it's not necessarily skeletal, but the one that's for glomerular. Yeah.
Yeah, okay. So your afferent and efferent arterioles are going to have almost equal hydrostatic pressure. So this is the pressure of the blood due to like the amount of force it's under. So your afferent arteriole has a higher hydrostatic pressure than your efferent, but only by about 2. And the reason why it drops a little bit is because you're filtering out some of that fluid. So if you're removing some of the volume of the fluid because it's entering Bowman's capsule, you're going to have a little bit higher, lower hydrostatic pressure as a result of that. But it doesn't drop too much. However, because you are removing some of that fluid from your capillary, the concentration of your albumin and your other proteins in the plasma is going to increase because the water is no longer diluting it. So your oncotic pressure is going to increase to promote the movement of water within the capillary. So it's going to want to hold on to more water because it's losing water, and so now your plasma proteins have increased in concentration, so they have a stronger hold on the amount of water to prevent it from leaving. However, unlike your skeletal muscle where your oncotic pressure or your hydrostatic pressure drops below your oncotic pressure, your oncotic pressure, even though it increased, it's still lower than your hydrostatic pressure. So you're going to still be promoting the movement of fluid out into Bowman's capsule. So it's going to promote filtration. So that's why it's different from your skeletal muscle, which once you get to the venous side, it's going to promote reabsorption. But we wouldn't really want reabsorption in the kidney because that would do the opposite of what we want. We want to move fluid into Bowman's space. So another thing to factor in is the hydrostatic pressure inside of Bowman's space. So with hydrostatic pressure, that's going to push fluid out. So if this is like higher than 15, if this has like a buildup of fluid for whatever reason, because of maybe like a blockage or something in the tubule, it's going to want to force fluid out. And that's going to prevent fluid from coming into the Bowman's capsule. And then also it has its own little oncotic pressure, but because your plasma proteins do not enter your Bowman space under normal conditions, then this oncotic pressure is 0 because there shouldn't be any proteins in here that would want to retain water in the Bowman's capsule. So it would be a pressure that wants to pull fluid into Bowman's space. So I kind of like drew this out here. This is the arrows of which direction you want the water to move. So your colloid osmotic pressure, this is going to be if there were any plasma proteins in your Bowman's capsule for whatever reason, like maybe you have like kidney damage and your plasma proteins are like entering the Bowman's capsule and like sticking in here, then maybe you would have this colloid osmotic pressure be something other than 0. So that would promote water out. Your glomerular hydrostatic pressure, this is the hydrostatic pressure inside the capillary that's going to want to push fluid out. Your capsular hydrostatic pressure, this is going to be if like fluid were to build up in your Bowman's capsule, that would want to move fluid outward and therefore prevent fluid from moving in. And then lastly, your colloid osmotic pressure, which is your plasma proteins in your blood that want to hold the fluid inside of the capillary. Does that make sense, or are you still confused?
I think it's just more of like maybe taking some time to understand, but it does make sense. I was just wanting to kind of like understand everything, but I kind of have to go over it like a little slowly, but I'll look at the drawing that you have as well.
Okay, yeah, let me
Just
know
make
if you
sure
have
it's like
any.
sticks in my brain, you know.
Yeah, it's a hard concept to like actually visualize, but yeah, let me know if you have any questions like next session or something if you're still confused.
Okay, thanks. I will. And just really quickly, one more time, you said that the hydrostatic pressure in Bowman's capsule increases or is higher just to avoid it from coming in?
No,
Avoid
so
water from coming in?
it's going to be low. It's only going to be 15. So your afferent and efferent are going to be like 60 and 58. It's going to be low because this pressure, sorry, I'll go back to this slide. This pressure is going to be greater than the hydrostatic pressure in Bowman's space, and that would allow it to overcome this pressure so that it can move fluid into Bowman's capsule, which is what we want. Does that make sense?
Oh, I think I was confused. I thought we were saying that we were forcing fluid out to prevent fluid from coming in.
Oh, so that would be like under pathological conditions. I'm sorry, I confused you.
Oh, okay. So if you had that's what you're saying, if it was like a bad kidney situation?
Yeah, if it was a bad kidney situation, this number would likely be higher and that would prevent fluid from coming in. But you want this to be lower than this so that fluid can come in because we want these pressures to want to promote fluid to enter Bowman's space so that you can like move it into the tubule and all that.
Okay, I see, I see. That way you can filter it and everything else.
Yes, exactly.
Okay. All right, sorry I confused that. Sorry.
No, I'm sorry I wasn't clear.
Phoebe, I'm a little confused on that because why is the purple then going into the capillary?
Okay, so for this is the pressure of the Bowman space. So if Bowman space had like fluid in it or more fluid than normal, it's going to want to push that fluid into here if it was greater than 60. So if it was greater than 60, then it would want to promote the movement of fluid back into the capillary because that pressure would be greater. But because it's only about 15, it's not going to like give much of a pressure difference. So it's not really going to do a whole lot. Does that make sense or is it still confusing?
Yeah, so I guess I'm just confused between the purple and the pink lines.
Oh, okay. Yeah. So your pink line is the hydrostatic pressure coming from the blood. So this is going to be that 60 in this slide here. So it's going to promote movement of fluid into Bowman's space. The capsular hydrostatic pressure would be this 15 here in your Bowman's capsule due to like, fluid accumulation in the Bowman's capsule. So because it's low, the pink is going to overcome the purple. So there's always going to be movement of fluid into Bowman's space. But if you had some kind of pathological condition that caused the hydrostatic pressure of Bowman's space to increase beyond 60, then it's going to want to promote fluid out into the capillary or into the interstitial space or whatever, but it would not allow fluid to come in.
Okay, that makes sense now. Thank you. Sorry about that.
No, it's okay. Any other questions, guys? You're welcome, guys, and email me if you have any other questions about it. I know this like last two lectures were a little bit confusing, especially with the PAH and the inulin. It's like hard to grasp, but I'll try to find some good videos on the countercurrent multiplier and the other one, the vasa recta and its role, and I'll post those because it's really hard to visualize otherwise. All right. Well, if that's the case, I guess I'll end the meeting and feel free to email me with any questions. Bye, guys.
Virgil got a paddock on my wrist going nuts. Niggas call me